Proteins digestion. In mouth, digestion of protein does not occur as there are no proteolytic enzymes in saliva. In stomach, pepsin, the important peptic enzyme of the stomach, is secreted by chief cells of gastric glands in an inactive form. Pepsinogen is then converted into pepsin by HCl. Pepsin is the most active at pH of range between 2 to 3 and is inactive at pH above 5. Therefore, HCl secretion by stomach is essential for the digestion of proteins. Pepsin acts on proteins and splits into proteases, peptones, and polypeptides. This splitting of proteins occurs as a result of hydrolysis at the peptide linkage between two amino acids. Gelatinase liquefies gelatin, and renin is absent in adult human, but it is abundant in infants, where it helps in the digestion of casein. Optimum pH of stomach is around 1.6 to 3.2. In small intestine, the protein digestion occurs mainly in the upper small intestine, that is in the duodenum and jejunum, under the influence of proteolytic enzymes from pancreatic secretion. Immediately on entering the small intestine from stomach, the partial breakdown products of the protein foods are attacked by major proteolytic pancreatic enzymes like trypsin, chymotrypsin, carboxypeptidase, and proelastase. Trypsin, chymotrypsin, and elastase, that is, these are the endopeptidases, act on the interior of the peptide chain to form small peptides. Elastases digest the elastin fibers. The carboxypeptidases or exopeptidases act on the terminal amino acids and produce small peptides into amino acids. Only a small percentage of the proteins are digested in the way to their constituent amino acids by pancreatic juices. Most remain as dipeptides and tripeptides. The last digestive stage of the proteins in the intestine lumen is achieved by enterocytes that line the villi of small intestine, mainly in the duodenum and jejunum. These cells have brush border that consists of hundreds of microvilli projecting from surface of each cell. In the membrane of each of these microvilli, there are multiple peptidases that protrude through the membranes to the exterior, where they come in contact with the interstitial fluids. Two types of peptidase enzymes are especially important: aminopeptidase and several dipeptidases. They succeed in splitting the remaining larger polypeptides into tripeptides and dipeptides and a few into amino acids. Both the amino acids plus dipeptides and tripeptides are easily transported through the microvillar membrane to the interior of the enterocyte. Finally, inside the cytosol of the enterocyte, there are intercellular peptidases that are specific for remaining types of linkages between amino acids. Within minutes, virtually all the last dipeptides and tripeptides are digested to the final stage to form single amino acids. Thus, the final digestion into amino acids occur in three locations: intestinal lumen, brush border, and cytoplasm of enterocyte. Digestion of nucleic acids. Nucleic acids are split into nucleotides in the intestine by pancreatic nucleases, and nucleotides are split into nucleosides and phosphoric acid by enzymes that appear to be located on the luminal surfaces of mucosal cells. The nucleosidases are then split into their constituent sugar, purine, and pyrimidine bases. The bases are absorbed by active transport. Transport and absorption. Most proteins after digestion are absorbed through the luminal membranes of the intestinal epithelial cells in the form of dipeptides, tripeptides, and few free amino acids. At last, 7 different systems transport amino acids. At least 7 different systems of transport of amino acids into enterocytes are there. So this multiplicity of transport protein is required because of the diverse binding properties of different amino acids and peptides. 5 of these require sodium and co-transport uh, amino acids and sodium in a fashion similar to co-transport of sodium and glucose. Two of these five also Two of these five also require chloride, and in two systems transport is independent of sodium. Dipeptides and tripeptides are transported into the enterocyte by a system that requires uh, hydrogen ion instead of sodium. In the enterocytes, amino acids are released from the peptides by intercellular hydrolysis, plus amino acids absorbed from the intestinal lumen and brush border are transported out of the enterocytes along their basolateral borders by 5 transport systems. 2 of these systems are dependent on sodium and 3 are not. From via hepatic portal blood, they reach the liver and general circulation. Significant amounts of small peptides also enter the portal blood.